Let's look at the internal structure of a skeletal muscle. A typical skeletal muscle is composed of an orderly arrangement of connective tissue and contractile cells. The entire muscle is surrounded by an external connective tissue wrapping called the epimysium. The skeletal muscle itself is made up of fascicles, which are bundles of individual muscle cells. Each fascicle is surrounded by a connective tissue layer called the perimysium. Click the fascicle to enlarge it. This view provides a closer look at the internal structure of a fascicle. Recall that fascicles are bundles of muscle cells. Within the fascicle, the third connective tissue layer, the endomysium, separates and electrically insulates the muscle cells from each other. All three connective tissue layers, the endomysium, the perimysium, and epimysium, bind the muscle cells together, providing strength and support to the entire muscle. The connective tissue layers merge at the ends of the muscle and are continuous with the tendons. To continue moving through the levels of skeletal muscle, click on the muscle cell to enlarge it and view its internal organization. Because skeletal muscle cells are elongated, they are often referred to as muscle fibers. Let's look at the internal structure of these unique cells. The nucleus is the cellular structure that contains the genetic material. The sarcolemma is a specific name for the plasma membrane of the muscle cell. The sarcoplasmic reticulum, or SR, is the name for the endoplasmic reticulum of the muscle cell. Its interconnecting tubules surround each myofibril like the sleeve of a loosely knit sweater. Terminal cisternae are sac-like regions of the sarcoplasmic reticulum that serve as specialized reservoirs of calcium ions. T-tubules are invaginations of the sarcolemma projecting deep into the muscle cell's interior. Note the visible T-tubule openings on the surface of the sarcolemma. A triad is a three-unit group consisting of one T-tubule lying between two adjacent terminal cisternae. The cytosol is the intracellular fluid in which the organelles are suspended. Mitochondria are cytoplasmic organelles that are sites of ATP synthesis. They are referred to as the powerhouses of the cell. A myofibril is a cylindrical bundle of contractile filaments within the skeletal muscle cell. To review any of these terms, click on the bold red words. Click the myofibril to enlarge it and explore its composition. The myofibrils that are packed into a muscle cell are composed of individual contractile proteins called myofilaments. There are two types of myofilaments. The thin filament is composed mainly of the protein actin. The thick filament is made up chiefly of the protein myosin. Details of thick and thin filament structure are covered in another topic, the sliding filament theory. To examine the arrangement of myofilaments, click the myofibril. The arrangement of thick and thin myofilaments forms light and dark alternating bands along the myofibril. This creates the cell's striated appearance, as shown here in this photomicrograph and diagram. Features of the light and dark bands are identified by letters. The dark bands are called A bands. The A band corresponds to the length of the thick filaments. The light bands, called I bands, alternate with the dark bands. The I band contains only thin filaments and corresponds to the distance between adjacent thick filaments. During muscle contraction, the I band gets narrower. Each I band is bisected by a zigzag line called the Z line. The Z line is actually a protein disc which anchors the thin filaments and connects adjacent myofibrils. In the middle of each A band, a lighter stripe appears called the H zone, corresponding to the region between the thin filaments. The width of the H zone varies depending on the degree of muscle contraction. It is widest when the muscle is relaxed and stretched. The M line, located in the center of the H zone, consists of protein fibers that connect neighboring thick filaments. The region of the myofibril between two successive Z lines is called a sarcomere. It is the contractile unit of a muscle cell. You will see how a muscle cell contracts in a later topic, the sliding filament theory.